Welcome to Module 3, everybody. I hope you're settling in well with the first two modules. This module will be all about circulation. And so not only are we going to talk about blood and blood vessels, but we're also going to get into the lymphatic system in this module. And then next module will be just focusing around the heart. All right, so blood is the theme of this lecture here. So remember that blood is a tissue, and remember a tissue is made up of multiple cell types. The matrix of blood is the fluid portion that we call plasma. And the cell types that are in blood form what we call the formed elements. And when we talk about formed elements, we're talking about leukocytes, which are white blood cells, erythrocytes, which are red blood cells, and then thrombocytes, which are platelets. Formed elements make up 45% of the blood composition, and plasma makes up 55%. Most of plasma is made up of water, but you also have these other dissolved substances such as gases, proteins, carbs, hormones, wastes. All of those types of things are also traveling in the plasma portion of the blood. So when we talk about blood, whole blood equals formed elements, so the cells, plus the plasma, which is the liquid portion. Human blood has a pH that is slightly alkaline or slightly basic at 7.35 to 7.45. And the volume of blood in an individual ranges from 4 to 6 liters. Women tend to have less volume than men, just because of our smaller stature, our higher fat content, and our lessened muscle mass. So this diagram is from your book. This just talks about what blood is made up of. So your formed elements plus your plasma. And this is about as far as we've gotten so far. Notice that plasma is made up of about 92% water. And then the next highest percentage is proteins, which include a lot of different things, clotting factors, antibodies are proteins, different enzymes, things like that. And then we will get into all of this, which are all of the different types of white blood cells. Those can be further differentiated out. But basically what I need you to know is that blood is made up of formed elements, which are the cells, the cellular component, plus plasma, which is the liquid component with all of the dissolved ions and substances. When you look at a blood smear under microscopy, I apologize, this is not super great, but this is a peripheral blood smear. So if you take a drop of blood, streak it out on a slide, microscope slide, and then look at it under microscopy, this is what you'll see. And looking at this, you see all of these pink reddish cells. Those are going to be your erythrocytes or your red blood cells. Those are the most abundant. And then all of these different purple cells are all different types of white blood cells. And we'll go into differentiating those from each other here in just a little bit. And then the smallest fragments, the smallest little cells here, you see this purple dot right here, that is a platelet. So when you look at a peripheral blood smear, the smallest cells are platelets. The medium-sized cells, the pink ones, are all red blood cells. And then the largest purple staining cells are all going to be white blood cells. This is all about the origin of our blood cells. So hematopoiesis would be the formation of new blood cells, and this occurs in the red bone marrow of the human body. And the precursor cell to all blood cells is going to be the stem cell, or a hemocytoblast. And that further differentiates into myeloid stem cells or lymphoid stem cells, and then those differentiate into red blood cells, platelets, and all the different types of white blood cells. But basically, this is kind of the area that I'm going to focus on here. And an erythroblast is the immature red blood cell, becomes a reticulocyte as it gets more mature. And then the reticulocyte leaves the bone marrow. Here's where everything's leaving the bone marrow here. And then your immature red blood cell or erythrocyte. Platelets are actually fragments of a cell called a megakaryoblast. So that fragments out and then that forms our platelets. And then there's lots of different kind of blast cells that eventually differentiate into white blood cells. But the ones that I'm going to focus on are going to be definitely the erythroblast and the megakaryoblast. So let's talk about red blood cells. They're biconcave, which you can see here. It's kind of like a donut shape or like an inner tube, but there's no hole through the middle, but it is sunken in the middle, which is that biconcave shape. You can even see it over here from both sides. That shape is helpful for red blood cells because they carry something called hemoglobin. And hemoglobin carries oxygen. So this shape of the red blood cell helps increase surface area to carry hemoglobin and oxygen, and also keep those things closest to the cell membrane. Mature erythrocytes lack nuclei and organelles, which means that they cannot reproduce themselves because they don't have any genetic material. The other interesting thing about that is they don't have mitochondria. So they do produce ATP, but not with mitochondria. They do it through like glycolysis, but they also do not use any oxygen that they carry. So that's handy, so that they're not actually utilizing oxygen that's located on them. Each red blood cell, we'll get into this a little bit more in detail, but each red blood cell can carry 4 oxygen molecules because there's 4 hemoglobin chains. The lifespan of a red blood cell is 120 days. And remember, it can't replicate because it doesn't have a nucleus. So new ones are being formed from the red bone marrow to replace the old. Remember from hormones, this hormone called erythropoietin. Erythropoietin is released mostly by the kidneys, but also by the liver to stimulate the red bone marrow to produce more red blood cells. So as the body detects that there's not enough red blood cells because maybe too many have died off, erythropoietin will be released into the bloodstream and then targets the bone marrow to make more. And this idea of reticulocytes I bring up because this is something that's commonly measured in lab, like blood work. Reticulocytes are immature, almost mature red blood cells. They have left the bone marrow, but they still may have a nucleus. The nucleus does not get ejected until the red blood cell is fully mature. This is something that's measured in labs a lot because if there is a large amount of reticulocytes being released into the bloodstream, then that means that you are super low in red blood cells and there may be a problem in your bone marrow releasing too many at once. Here's that negative feedback mechanism. I think I maybe showed this last week, but this is just illustrating a time in which we might need more red blood cells. So for example, the stimulus here would be our blood oxygen content is low. And if our blood oxygen content is low, that means that we maybe don't have enough red blood cells to carry oxygen, or they're not doing a good job carrying oxygen, which is another medical issue. But let's just say that our blood oxygen is low. Let's say that we're going on a hike up a mountain somewhere, higher altitude, so our oxygen level is lower. What's going to happen is our liver and our kidney are going to release this hormone called erythropoietin into the bloodstream, and that's going to travel to our bone marrow. This one is in the femur, and stimulate the production of red blood cells released into the bloodstream. Now, as we increase the number of red blood cells, we are increasing the amount of oxygen that can be carried throughout the body. And as oxygen increases back to normal or a normal set point, now we're going to shut off that erythropoietin because we have now reached homeostasis. So clinical correlation here that we may talk about is anemia. So this is there's different types of anemia, but one might be a deficiency of actual red blood cells, so we don't have enough. But maybe we have a reduced amount of hemoglobin on those red blood cells, and those red blood cells, we might have enough, but they don't have the capacity to carry the oxygen that our body needs. Common symptoms, this is not a full list of symptoms by any means, but two very standout symptoms would be pale color. Hemoglobin is red, or gives us our like pink color in our blood. And so the less hemoglobin that we have, the more pale we're going to be. And then also just feeling fatigued. The less oxygen that you have, the less you can move your muscles and recover from that. And maybe shortness of breath would even accompany that as well. So different causes, just depending on what type of anemia you are looking at, maybe decreased erythrocyte number. And so this would be a lack of red blood cells total. Maybe because we're bleeding out somewhere, right? You have a really bad injury and you're losing a lot of blood. Certain types of infections can also kill off red blood cells. Lack of ability to absorb vitamin B12. Vitamin B12 is necessary for iron production and that hemoglobin capacity. And so if we cannot absorb B12, then we're not going to be able to have mature red blood cells that are capable of carrying oxygen. Or maybe there's a problem in our bone marrow. Maybe our bone marrow is simply not able to produce enough red blood cells for whatever reason, maybe like a cancer or something like that. Another type of anemia would include decreased hemoglobin. This might be dietary related, so a lack of iron. Hemoglobin, we'll talk about it a little bit later, but one component of the hemoglobin molecule is iron. And so if we don't have enough iron, we cannot make enough hemoglobin to carry oxygen. Or again, persistent bleeding in terms of like maybe like an intestinal bleed or an ulcer or something like that. Or maybe we have hemoglobin, but it's got a genetic abnormality in it to where it's not able to carry oxygen efficiently. These would be like your factor diseases and like sickle cell anemia, things like that. Oh, I'm going to go back here. One thing I did want to point out is these red blood cells. So a normal red blood cell does have kind of a faint white center, and that's just from that biconcave shape. So basically what you do is you shine a light through these red blood cells, right? You smear them on a slide and then you have light from your microscope coming through the bottom, and you're looking through, shining the light through it to be able to see it. So you will naturally have like a lighter center here because they are that biconcave shape in the middle. But these ones have a very large center, very, very, very pale red blood cell. And remember that hemoglobin gives the color red. And so the less hemoglobin that you have, the more white and the largest center you're going to have here on your cell. So hemoglobin. Hemoglobin is a molecule here that's made up of 4 globin chains. Those are protein chains or polypeptides. And then each polypeptide chain or globin chain has a heme group on it, which contains iron. Each of these units can carry 1 oxygen molecule. So each red blood cell has the capacity to carry 4 oxygen molecules. They also carry a little bit of carbon dioxide, and we'll get into more of that when we get into the respiratory system. So our red blood cells only live for 120 days. Then what happens? They are naturally in circulation, and so they will naturally travel to the spleen or the liver. The spleen and the liver have checks in place to destroy red blood cells that are maybe no longer functioning or not carrying the oxygen that they're supposed to or something like that. So there are like macrophages and things like that in these areas that will degrade red blood cells that are no longer functioning or functioning appropriately. So the cell breaks down, we recycle the hemoglobin. So there's 4 hemoglobin molecules on each red blood cell. The heme group here, these little guys, will break down into iron plus a pigment we call biliverdin. The iron will be transported to the red bone marrow. Transferrin is a transporting molecule that will help iron go through the bloodstream. So it's kind of a helper. So iron will bind up to transferrin, and that will go off to the red bone marrow to be put onto new red blood cells. Most of it, though, does not go to the red bone marrow. Most of it is stored in the liver as an iron storage molecule called ferritin. Ferritin and transferrin are common things that you measure in a blood test if somebody is suspected to be anemic, so that we can figure out if it's a problem with having too little iron binding molecules in the blood, or we don't have enough stored in the liver, or what the actual problem is, so that they can diagnose that anemia more specifically. This pigment called biliverdin breaks down into a pigment called bilirubin. And this is another pigment that is measured in blood, especially in gastroenterology for liver patients. And those are excreted in bile. So bilirubin, if you've ever seen somebody that's jaundiced, right, they have that kind of yellowy color, that would be potentially caused by an excess amount of bilirubin. And that could go back to the excess breakdown of red blood cells, which means that the liver is malfunctioning somehow because it shouldn't be killing off all of those red blood cells. So very interesting things here. The fate of red blood cells is actually super related to the liver. So a lot of cool things to kind of look at here. So that's what happens to heme. The globin chains break down into amino acids and are just recycled, because remember, those are proteins, which are long chains of amino acids. Here's what this looks like. This is a picture from your book. So let's start here in the small intestine, which is where your B12, folic acid are all absorbed from what you eat. Those things are carried to the bone marrow to make more red blood cells, which are released out here into circulation. And here is where the fate of a red blood cell would occur. So here's old red blood cells here. Hemoglobin is broken up into globin plus heme. Heme is further broken down into iron and biliverdin. Globin is broken down into its amino acids. That biliverdin breaks down into bilirubin and then goes out the bile tract, which we'll get to more in digestion. But basically just kind of knowing the components here is the goal for right now. See here. Okay, so let's get into white blood cells. So white blood cells are called leukocytes. White blood cells have an immune function. They differentiate in response to hormones called interleukins and colony stimulating factors. So unlike red blood cells, which respond to the hormone erythropoietin, leukocytes are going to respond to different types of hormones here. They are still made in the red bone marrow. They mature in your immune glands, which we're going to get to lymphatic system, some in the thymus, some migrate to like lymph nodes or the spleen, or let's see, lymph nodes, spleen, thymus. That would be it. The interesting thing about white blood cells is they have the ability to actually leave the bloodstream. So red blood cells aren't supposed to be leaving the bloodstream because that would cause bleeding in our tissues. But white blood cells need to be able to leave the blood vessel to be able to fight infection in our surrounding tissues. And that ability to leave the bloodstream or squeeze through the cells that make up our blood vessel is called diapedesis. And why would they leave the blood cell? Well, they are sent signals from damaged tissue. So let's take this an example. We get like a sliver or something penetrating our skin, right? That's going to naturally break a lot of blood vessels, capillaries in here. Our white blood cells can exit those capillaries to the surrounding tissue because now we have bacteria on that object that just penetrated us. That bacteria is going to spread to all of the surrounding skin tissue. And our white blood cells will leave the bloodstream here to go to the surrounding tissues to kill that bacteria. And how does it do that? Positive chemotaxis, which is a phenomenon in which these cells that have all been injured or affected by bacteria release something called histamine, or other chemicals, and that signal or attract white blood cells to come to that site. So that's called positive chemotaxis, responding to a chemical release of some sort to kind of attract our immune cells. It's kind of like, I think about like bats and echolocation, right? So bats let out like this echolocation, like ultrasonic wave, and that attracts like bugs or whatever for it to eat. So that's kind of like what I think about when I think about positive chemotaxis. Leukocytes can be split up into two groups, granulocytes and agranulocytes. What you'll notice, the biggest difference is going to be their cytoplasm. So granulocytes are named because they have granules in their cytoplasm, or little dots. We'll see that. Agranulocytes don't have those. Granulocytes also have a shorter lifespan. Agranulocytes live longer. Then the 5 different types are split up into these groups. The fills, I like to call them, are granulocytes. And then the agranulocytes are going to be the oocytes. We'll get into that. Granulocytes are going to be neutrophils, eosinophils, basophils, lymphocytes, and monocytes are going to be here in the agranulocytes. So let's talk about the granulocytes. The first one that we're going to talk about is neutrophils. Neutrophils are the most common white blood cell in the body. They make about 50 to 70% up of our white blood cells at any given time. They look like this. So here's its weird shaped multi-lobed nucleus. This one actually has 4 lobes, 1, 2, 3, 4. This is the nucleus, multi-lobed. And then these granules here stain usually light purple or pinkish. So that's why we call it a granulocyte there. These are the first responders to a site of infection, mostly bacterial infection, and they are good because they can perform phagocytosis, which is basically the engulfing of bacteria. I think of Pac-Man when I think about that, these little guys that go through and then eat bacteria or infected cells or something like that. So these guys are the most abundant, and I would say the most important. The next granulocyte are going to be eosinophils. There's not very many of these circulating at any given time, only about 1 to 3%. The textbook feature here are going to be these very dark red, or sometimes they just look pink, but they're very dark and prominent granules in the cytoplasm. The nucleus is usually bilobed, so usually 2 lobes. It usually looks like a horseshoe. But eosinophils respond to like allergic reactions and parasitic infections. Basophils make up less than 1%, very rare white blood cell here. They have a very irregular, deep granular cytoplasm. And the nucleus here, it's here, but it's usually obscured by all of these huge dark granules. So there is a nucleus here. It's hard to tell how many lobes it has or anything like that, but these very dark, large granules are kind of textbook about basophils. Basophils release heparin and histamine at the site of damage. Now, what does heparin and histamine do? Heparin is like what you would think of as like a blood thinner. And so heparin is going to prevent blood clotting. So let's say that you have that injury, right? And heparin is going to be released to actually promote bleeding here for a little while. That's just to get these leukocytes to the site of infection. We don't want this blood to clot off because we need to get those leukocytes to the site of infection. The other thing that it releases is something called histamine. Histamine promotes inflammation, and inflammation is important because it can help stop the spread of infection, actually. So inflammation is actually a good thing. It's a important thing when we talk about hemostasis or like blood control, infection fighting, things like that. Inflammation is a good thing. And so inflammation not only stops the injury from spreading over to these other tissues, But inflammation also makes the blood vessels more leaky or permeable. And so more of those white blood cells can squeeze through the cell membrane here of a blood vessel. Okay, so those were the granulocytes, those were the fills. Now the cytes are the agranulocytes, monocytes. Those make up 3 to 9%. I know that this cytoplasm looks kind of granular, but if you compare it to these other ones, it's really not. It looks more smooth. The nucleus of a monocyte changes shape morphologically. Usually it's this kidney shape. Sometimes it's more of a horseshoe shape. But the thing you're looking for monocytes is basically dark purple and light purple. And then what does the nucleus look like? Because that will differ from our other agranulocyte. So monocytes are necessary to transform into macrophages. So they are those little Pac-Man guys that go through the bloodstream and chomp up cellular debris and infectious agents. And they respond mostly to viral infection and sites of inflammation. So if you have that injury by that splinter, there's going to be monocytes there responding right away. The other one is a lymphocyte, and these are the second most abundant leukocyte in the body. They are also the smallest. Now, the nucleus here, we have dark purple, light purple again, just like the monocyte, but this nucleus is very large. It takes up almost the entire cell, and it's very, very dark. And it's spherical. It's not this kidney or horseshoe shape. And these little guys are what's going to differentiate into cells that we know as T cells and B cells. We're going to talk about more of this when we get into that lymphatic lecture. But T cells and B cells are going to be our major cells that are responsible for immunity and memory of pathogens. Thrombocytes are platelets. So again, they come from megakaryoplasts that are broken up into little fragments called platelets. These are the smallest formed element in the bloodstream. They have a cell membrane. They also have lysosomes and mitochondria, but they do lack a nucleus. So they have some organelles, but they do lack a nucleus, which means they cannot reproduce themselves. They only live for about 10 days, and then more of these megakaryoplasts will split up. They release serotonin. Serotonin is a neurotransmitter or hormone, just depending on where it's released from, that promotes vasoconstriction. And so when you have that injury, we're going to talk more about hemostasis, but when you have that injury, we want that blood vessel to constrict right away to prevent bleeding to other areas. In terms of like major vessels, right? So these small vessels will constrict and hopefully stop a hemorrhage on their own. We'll talk about that when we get to hemostasis. They also stimulate fibroblasts. Fibroblasts rebuild tissue that's injured on the blood vessel wall. And so that blood vessel wall has been cut or damaged, so we need to rebuild that epithelium. And so fibroblasts are going to come in and do that. Okay, hemostasis. Hemostasis is the stoppage of bleeding when vessels are damaged. This is another example of positive feedback. So the major example of positive feedback that I've been giving is childbirth, which is a very good one. But this is also one that you may not think about. The first step would be vascular spasm. The second step would be a platelet plug formation. Third step, blood coagulation. And then finally, we're rebuilding the fibers, the collagen fibers of our blood vessel wall. So our first step, when a blood vessel is damaged, that stimulates a spasm. It's a reflex. So breaking a blood vessel, we want that blood vessel to spasm. And so it's going to stop the bleeding into the tissue. Now, remember, we still want to promote blood flow into that area. So everything kind of above that isn't going to spasm, just that site of infection to lessen the flow of blood into the tissue. This is a very short-lived response. This is just an initial reflex, very quick action. But basically the idea again is to reduce as much blood loss as possible where that blood vessel is broken. Then what happens is our platelets are going to come in, our thrombocytes, and they are going to come in and plug up that area. So the spasm happens, the spasm is very short-lived, so it may or may not stop the bleeding completely. Okay, so here's our blood vessel that's been completely broken. We have these little platelets here. See all the red blood cells that are exiting there. It's escaping. We don't want that. We want to stop that. So this wall might spasm, but it's not going to last forever. So our platelets are going to come in and they're going to plug up that hole. And as those platelets build that wall, now we have stopped the blood flow. We still have blood flow coming in from this way though, because we need to deliver white blood cells and things like that to the site of injury to prevent infection. Then what happens is blood coagulation. So the platelet plug is a good temporary hold, but then we need to actually make a blood clot. And the difference between a platelet plug and a blood clot is the use of fibrin, which are these little, they're yellow in this electron micrograph, but these fibrin fibers are reinforcing that platelet plug to make it stronger and more mesh-like, like a net. How does fibrin get there? Well, we have something circulating in our blood called prothrombin. And again, for those of you who are going into the medical field, prothrombin is one of those things that is tested a lot in blood work for people. The more prothrombin that you have, the more at risk you might be for converting fibrinogen to fibrin or forming a clot. And so that's something to just keep an eye on. Prothrombin gets activated into thrombin by this enzyme, this prothrombin activator. And then thrombin stimulates the conversion of fibrinogen to fibrin, which is inactive fibrin to active fibrin. So a very interesting stepwise pathway here. It starts with this prothrombin to thrombin and then fibrinogen to fibrin. Prothrombin is something that we test in people who are on blood thinners a lot, just to make sure that those blood thinners are working. Clotting factors, also your factor, you've all heard of factors, right? Factor 9, factor 10, whatever it is. Those things are also involved in this. Your book goes into a lot of detail about that. I'm not going to. I'm more concerned just about this simpler area first. It does get very complicated when you start bringing in all those different factors in. But if you want to know more about those factors, just take a look at your book reading. And blood clotting can be broken up into two different types. We've got extrinsic and intrinsic. Difference would be, are the blood vessels broken or not? So extrinsic would be a broken blood vessel or damaged tissues. Usually an external injury of some sort caused this to happen. Intrinsic would be the absence of this, but we still have a blood clot forming. I'm going to get into more of that here in just a minute. So the last step here after that blood clot is formed is that we need to rebuild that vessel wall, and so that our circulation can be restored to its normal. So what's going to happen is the fibroblasts, which are cells that promote like collagen synthesis, are going to form new connective tissue to make that blood vessel wall, and clots are going to dissolve so that we can restore blood flow to that area. It's kind of like building a new bridge. And we have something called plasmin that's going to kind of digest that clot when this starts occurring. Now, an abnormal blood clot in a vessel is called a thrombus. And when that thrombus becomes modal or mobile and travels somewhere else in the body, it's called an embolus. So a lot of times in the medical field, you have somebody who maybe doesn't move a whole lot, somebody who's bedbound, they're at risk for forming blood clots in like the leg. And we worry about that because we don't want it to travel to the lungs. Naturally, circulation, everything travels back to the heart. And so if we have a thrombus in our leg and it breaks free and it is now mobile, it could travel to the lungs, causing an embolus, which is very dangerous. Extrinsically, so again, this is a response to damaged or broken blood vessels. Again, prothrombin is converted to thrombin, which stimulates fibrin formation. So the more thrombin that you have, the more prothrombin activator that you have. And then that's the positive feedback mechanism. So this is the blood clot formation. This is the positive feedback that I was kind of talking about. So prothrombin is converted to thrombin via this prothrombin activator. So the more thrombin that you have, it positively feeds back to the prothrombin activator here, which causes, again, more thrombin to be formed. And so bleeding or hemostasis, bleeding happens, and then you just keep building and building and building and promoting blood clotting until you reach the end point of that clot, to where the end point is our bleeding has stopped. And that's the positive feedback mechanism of blood coagulation. So normally in a normal person, why don't you form blood clots all the time? Because all of these things are circulating in your bloodstream all the time. We also have something called antithrombin that prevents it. Our blood vessels are naturally very smooth on the inside. Fibrin likes to stick to jagged and rough surfaces, like if your blood vessel was broken, or if you have a lot of plaque buildup in your vessel wall, that's something that fibrin can stick to as well. But normally in a healthy individual, the inside of our blood vessel is very smooth. And so fibrin and platelets and things don't like to stick to that. And then we also have this antithrombin, which is like a natural anticoagulation molecule. Now, intrinsic mechanism would be not needing to damage a blood vessel in order to form a clot. And this is just the exposure to a foreign surface. So again, the blood vessel wall is very smooth in a normal, healthy individual. Well, what about somebody who has maybe like atherosclerosis, where they have a lot of plaque buildup in their arteries, or their arteries are a little bit harder texture than normal? That's going to be attractive to fibrin and platelets to want to stick to that. And that is how a blood clot occurs most commonly in an individual without actual damage of a blood vessel. That's intrinsic. And examples would be like a stroke, right? Or a heart attack, where a clot is occluding a circulatory pathway and then blocking blood flow to a certain area of the heart or a certain area of the brain. And then the lack of blood flow equals lack of oxygen being delivered to those tissues, and tissues need oxygen to survive. And so you would have tissue damage or tissue death from that. Okay, so with that, I am going to break here. And when we come back, we will focus on we need to talk about blood typing. So that will be in the next lecture.